My name is Gina, and I'm a graduate student at the University of Wisconsin-Madison in the microbiology department in the lab of Cameron Curry. And I study how microbes and communities of microbes in the environment are able to degrade cellulose. And so the main organisms that we study are leafcutter ants. Leafcutter ants are the dominant herbivores in Central and South American rainforests, and they take in huge amounts of plant biomass, up to like 500 kilograms of leaf material in a year. But what's really, really cool about leafcutter ants is that they don't eat any of the leaf material themselves. They are also called fungus farming ants. So they take the leaf material and they feed it to a fungus that they farm. And the fungus is able to produce all the enzymes necessary to break down the biomass. And so the fungus degrades the leaves, and then it produces special structures that the ants can eat. We can study the fungus and understand how the fungus is breaking down the leaf material, and that will give us clues on how we as biofuels researchers can break down leaf material better and make biofuels more sustainably and cheaply. Bioprospecting is when you go and you look in the environment for some sort of system, some sort of product, something that's desirable that is useful to humans. In the case of biofuels, we're interested in going and bioprospecting to find organisms and enzymes that are really good at breaking down biomass. The test tubes with the filter paper made from cellulose that the students are setting up in the lab is pretty much the exact same setup that I use in my research. I do the same thing. I go out into the environment, I collect samples that I think will be good at degrading cellulose, and then I take a tiny little bit of that sample and I put it into the test tube, and I compare lots of different samples to see which one's the best, and then focus my efforts on understanding what's going on in that best sample. The GLVRC wants to identify the best, most efficient, most effective way to degrade cellulose. And so if students are able to identify that soil or that cow poop or that rotting logs are always really, really good at degrading cellulose, then we can say, wow, we should really be going and trying to isolate organisms and understand the enzymes in that specific type of sample. We're interested in that because by understanding how these environmental organisms can degrade cellulose, we can learn about how to better degrade cellulose ourselves for biofuels.